Embryology is a study and analysis of embryos. Evidence of an evolutionary common ancestor is seen in similarity of embryos is markedly different species. Darwin used the science of embryology to support his conclusions. Embryos and the development of embryos of various species within a class are similar, even if their adult forms look nothing alike. It's a branch of comparative anatomy which studies the development of vertebrate animals before birth or hatching. Like adults, embryos show similarities that can support common ancestry. All vertebrate embryos have gills, slits, and tails. They connect the throat to the outside early in development but eventually close in many species. There are three branches when talking about embryology: fish and larval amphibians, which they contribute to the development of gills; mammals, the tissue between the first gills slits forms part of the lower jaw and the bones of the inner ear. The embryonic tail does not develop into the tail in all species. Humans, it is reduced during the development of the coccyx or tailbone. Similar structures develop during support common ancestry. So, when talking about the branch of embryology, scientists in the field have added to our knowledge of the human body by discovering, for example, that there are three major embryonic categories of cells called the germ cell layers in our bodies. The layers are ectoderm, endoderm, and mesoderm. Ectoderm forms the epithelium, the thin tissue that creates the layer, which is the outside of the body surface and lines of the alimentary canal and other hollow structures, which not only covers the body but also gives rise to cells in the nervous system. Endoderm, it forms the gastrointestinal tract and associated structures involved in digestion. And mesoderm forms a connective and soft tissue such as bone, muscle, and fat. And after birth, some cells in the body continue to proliferate while others don't and remain or are lost in aging process. Aging results from the inability of cells to maintain or replace themselves. So when talking about mammals in general, the process is quite long, but the first thing is mammal develops from a single cell called zygote, which results from an ovum being fertilized by a single sperm. The zygote is surrounded by a strong membrane of glycoproteins called the zona pellucida, which is the successful sperm that has managed to penetrate. The zygote undergoes cleavage, increasing the number of cells within the zona pellucida. After the 8-cell stage, mammalian embryos undergo what is called compaction, where the cells bind tightly to each other, forming a compact sphere. So caviation occurs next, where the outermost layer of cells, the trophoblast, secrete water into a morula. As a consequence of this, when the number of cells reaches to 40 to 150, a central fluid-filled cavity has been formed. The zona pellucida begins to degenerate, allowing the embryo to increase its volume. The stage in the developing embryo reached after 4 to 6 days is the blastocyst and lasts approximately into the implantation in the uterus. The blastocyst is characterized by a group of cells called the inner cell mass and the toroplast. So this is really similar to the embryology of humans, since humans are mammals.